How are the weekends? They're still underway, it sort of feels like. Yeah, that's a week from this Friday.
Yeah.
Okay. Yeah. All right. So we have Monday and Wednesday this week, and then we have a really long break. Every time there's a break of one extra day, it doesn't really have any recuperative power. All it does is slow momentum so that Monday strikes harder. To me, that's what it feels like to me. The following Monday. I mean, I'll take it every time that there's one of these breaks. I'll take it. And I'll liken the situation to Mike Tyson getting out of prison and being unable to take a punch. All right, so we're moving into hypertrophy. We talked about protein catabolism a couple lectures ago. We talked about protein anabolism, sort of broad perspective last lecture. And now moving into more of the application and a little bit deeper into what anabolism looks like, hypertrophy, how that works, and so how you actually transition, make your body recognize the stresses that are put upon it. I messed up on some of the points, I corrected them, so those are all updated, and everything is here, you know where to find stuff. So the case study that's obviously coming soon, we did Oprah, we did the weight loss one, and there's a weight gain one that's coming soon, a team competition of weight gain, and I don't know exactly what day we'll do it on, but it's coming soon. I want to get through enough of the mTOR and hypertrophy material so that you have something to draw from. So somewhere around the end or shortly after we finish all of the hypertrophy stuff, we'll talk about using that, practical applications of all that material. But always start with this stuff. Always start with the grading of somebody, the risk factors, how risky a scenario this is. You know about metabolism, catabolism, anabolism of fat and carbs and protein, catabolism of fat and carbs and protein, where those amino acids go. And then also leucine and lysine, they have a little bit different fate. There's the amine group, the nitrogen part, the amine group, you get rid of that, you use it as Krebs cycle intermediates mostly, not exclusively, but a lot of that. Remember the phenylalanine to tyrosine conversion, because this is going to come up probably later if we're talking about mechanisms of fatigue or if we're talking about, actually I think we'll do it right before we leave for Thanksgiving break. We'll talk about tryptophan. And so the phenylalanine, large neutral amino acid transporter phenomenon will make a resurgence. It will come back. The way we degrade our proteins, the way we break these things down, the lysosome is going to make a bigger and bigger appearance as we talk more and more about mTOR, as the lysosome is where mTOR shows up at the work site. That is the work site for mTOR, which is the major way we're regulating protein translation. So this will make a bigger appearance. You know the proteasome stuff. The proteasome, we are degrading specific proteins, or the E1, E2, E3 stuff. We're attaching ubiquitin to this protein. Get 4 of those ubiquitins, run it through the gantlet was the word I was trying to think of. You run it through the gantlet and degrade all of that, the proteasome, degrade all of that protein. So E1, E2, E3. And you know the AMP, activated protein kinase, AMPK, phosphorylates a ton of things. It's a kinase, so it phosphorylates stuff. That's what kinases do. FOXO, forkhead boxo, that's one of them. And downstream from FOXO, AMPK, FOXO, we see the lysosomal system, protein degradation, the ubiquitin proteasome system, and this promotion of catabolism, a promotion of atrophy, the loss of protein downstream from AMPK activation, but we don't degrade all proteins similarly, right? There are different paces of degradation. There are some proteins we let go of with relative ease compared to some of those longer lasting proteins. And ubiquitin proteasome system, we can activate ubiquitin and the proteasome separately. In the case of starvation, we're going to activate them both simultaneously. And when those are functioning in tandem, you are very degrading, right? You are very atrophy oriented. But it just, as you know, you can activate the proteasome independently, forskolin, glucagon, epinephrine. Let's get all of that PKA signaling. So PKA is sort of the opposite of mTOR. PKA signaling is, and you know how this works, right? The pump. You know what the pump is in exercise? You go to the gym, you do whatever steps you're doing, and that muscle acutely swells. You get a bunch of fluid in there. There's an acute swelling of that muscle. It's called hyperemia, exertional or functional hyperemia, reactive hyperemia. As long as the word hyperemia appears, a bunch of blood flow going to the muscle is what's causing it. And remember that capillaries are not constricting and dilating. Those capillaries are tiny. Those things are not constricting and dilating. If you want to send more blood to a tissue, remember metabolism drives blood flow. Metabolism is what's going to determine where your blood flow is going. And if you want to send more blood to a tissue, you have to dilate the upstream arterioles, the upstream branches. You can't dilate these things. And a single muscle cell is going to be longer than the capillary supply. So usually multiple capillaries are going to supply nutrition for a single cell, for a single muscle cell. And those capillaries may be coming from different upstream arterioles. So one muscle cell may be causing dilation in multiple upstream arterioles. The amount of total blood flow going to the muscle during exercise can be enormously overshot, right? It can be this, right? The amount of blood flow can be enormously overshot. And the muscles determine that. What the muscles are doing in releasing these metabolic byproducts, what those muscles are releasing, are telling those upstream arterioles how to behave. Hey, I need more nutrition, so please dilate something before, you know, my capillary bed, before my little neighborhood. So there's lots of stuff that can cause it. Remember with hypertrophy, which is totally different. Now, having a big pump, there's some evidence that it's going to contribute to a hypertrophic response, both in the Florian Lang cell swelling stuff and in the stretching of the tissues. So having a pump contributes in a couple of ways, likely magnitude, I don't know, but contributes to the hypertrophic response. But the hypertrophic response itself, at the onset of exercise, is missing. At the onset of resistance training, you don't get a hypertrophic response. I mean, it's almost completely missing. Workout after workout after workout after workout after workout, you don't get it. What you get is neuronal adaptations. Remember rate coding, Golgi tendon organ stuff, agonist, antagonist relationships, things like that. GABA, stuff like that. And then once it does set in, eventually you're like, all right, enough neuronal adaptation. Let's get some actual protein accretion. Let's get some bigger tissue. You'll grow pretty well for a while, and then you'll level off. You'll grow for a while, and then the trainability principle ensures that you start slowing down, because with each additional gram of protein you gain, with each incremental improvement in a cell, there's less total improvement to be had. don't know what to compare it to. I mean, but just with like skill acquisition, it's the exact same thing. Trainability principle applies to any division of fitness, any component of health and fitness and capacity. So if it's a sport, we talked about tennis last time. I'm going to develop very quickly in my skills, but I'm going to slow down the better I get. There's just, you have less and less improvement left in the bank to draw from, and you don't take it all at once. So you get these incremental improvements slow down. Eventually people will hit ceilings, but when people think they've hit a ceiling, almost never the case. Really what people need at that point is better training principles. You know, if it's exercise, better exercise biochemistry applied. So the types of hypertrophy, right? Remember there's hyperplasia, which is the increase in the number of cells, and there's hypertrophy, which is the increase in the cell size. Hypertrophy is a matter of protein deposition. You're increasing the amount of protein in an individual cell. Hyperplasia is more cells. Both exist, but the bulk of training adaptation is hypertrophy. It's more and more protein within that cell. Now the proteins that get deposited, they're not all actin-myosin. The proteins that find themselves inside of a cell, it's not just functional cross-bridge cycling actin and myosin. There's other stuff too. And the nature of training can influence the nature of protein deposition, right? Which proteins go, is it just girder structure? Is it a bunch of connective tissue? Is it this sarcoplasmic growth without actin and myosin? Or is it functional training? Power weight ratio kind of functional training. The muscle doesn't weigh that much, but it sure can produce a lot of force. Those are two different responses to two different types of training. Remember the cell swelling stuff, that pump, hydration, dehydration has implications. If you're training in the heat and you're not replenishing those fluids effectively, if you're getting dehydrated, you are diminishing anabolic signaling. So for a case study, if somebody's in California and they're exercising, they're lifting weights outside or something. Oh, it's summer, I'm outdoors, I'm lifting weights. Oh, hydration becomes key. And if you're an athlete, hydration is key, right? I mean, just like, wet your pants if you have to. Like, to make sure those cells are hydrated. Otherwise, why are you exercising at all? Right? There's no point in exercising for the sake of exercise. Right? That's a indulgence in masochism. What you want to do is exercise for the purpose of improvement, exercise for the enhancement that it permits. And if you're exercising while dehydrated, Florian Lay is looking at you with a suspicious eye. It's not going to be as helpful. Hypoxia was another trigger, just like cell swelling is a trigger. Hypoxia, low oxygen, hypoxygen, is another trigger for hypertrophy. And there are techniques. You know, we talked about you can get that tourniquet around your arm, occlude some blood flow, and keep exercising with a lower load, and you'll get similar cell signaling as you would without an occlusion and the higher load. Nutrient availability changes, right? What do you have on board that's to use metabolically in the presence of hypoxia? It's not a bunch of fat. Yeah, a bunch of carbs. Yeah, use a bunch of carbs and get a bunch of lactate built up, and then lactate's association with growth hormone, and you start looking at some of this MAPK, microgen activated protein kinase signaling. You see the response to training in the presence of hypoxia, and it's similar to training at a high load, training at a more intense load. Because intensity, the more intense you get with exercise, the more hypoxic at the cellular level that becomes. At the tissue, at the level of the exercising tissue, the higher the intensity, the less oxygen there is available to supply all that intensity. So hypoxia is sort of a surrogate assessment for intensity, and it shows when you go into cell signaling. Now, this is what we're really going to focus on, the myogenic pathways. And you see AKT, that's PKB, that's the same thing. That's just two different names for the same thing. PKB and the mammalian target of rapamycin, mTOR. So mTOR, MAPK, mitogen activated protein kinase, and stuff like calcineurin. We'll talk less about that. Calcium is that double-edged sword a little bit. But getting more calcium into the cell during exercise does stimulate some anabolic pathways. So we'll talk a little bit about that. But MAPK and mTOR, these two pathways of protein synthesis, of remodeling a damaged tissue, are really the governors of the whole thing. And people will talk about satellite cells and hypertrophy, and they're important. Satellite cells, super important. They live right here in this little space between the basal lamina and the sarcolemma. They live right there. And then when they get activated, they're quiescent, right? They're dormant. They're alive, but they're napping. They're sleeping. And then they're activated, and you build them up, and then they fuse, right? They donate their parts. You can make a new fiber out of these, line those things up, and there's a bunch of nuclei in them, but they can contribute their nucleus, they can contribute their components to the existing fiber. So there's some hypertrophy right there. And the functions of these satellite cells go beyond. They're also expressing these myogenic regulatory factors. And so these transcriptional regulators, like gene expression. What are your genes doing? Not what genes exist. What genes do you have? Go to your 23andMe, but what are your genes doing? How are they expressing themselves? And these satellite cells express a lot of these regulatory factors. But the problem is, mTOR is pretty much how you're stimulating them anyway. So if you want to interact with this in a functional way, these are both from the same article. You'll see it online. And so if you're going to line these things up and fuse them and make muscle fiber, okay, that's how you do it. You see MyoD, you see myogene and MHC, that's myosin heavy chain. And down here, this is that hypertrophy model. But what you see is Wnt signaling. You get more and more and more Wnt proteins doing the signaling. And I'll get to that toward the end of today, what this Wnt signaling actually is to make these things behave in that way. You'll figure it out immediately with this one, and this article will be up also later today. But mTOR is necessary for satellite cell proliferation and differentiation. So do you want your satellite cells contributing to hypertrophy? Do you want the repair and remodeling of your tissue, the regeneration of damaged muscle tissue from exercise or injury, whatever? If you want that recuperation via satellite cells, you need mTOR to trigger that, mTOR to signal that. Deletion of mTOR leads to reduced expression of key myogenic genes. So all of that, whatever your satellite cells are doing, if you just work with mTOR, that's how you engage. Now you need appropriate loading, stuff like that. We'll talk about that. But mTOR ends up being this major regulator of all of this. More important than any of its peers. And even when we look at like cell swelling, well, how does cell swelling enact anabolism? Well, it's a bunch of like stuff for mTOR. mTOR ends up being really key to this stuff. How does hypoxia enact its anabolism? Well, all of those metabolic byproducts that get released in excess with hypoxia trigger mTOR. So it all goes, not all of it, but the bulk of it, most anabolism, most protein synthesis is going to go through mTOR. If you knock out mTOR, you knock out all of those pathways. None of those things end up working for you. And so this is looking at those transcriptional factors. Rappa is rapamycin, that's a mTOR inhibitor. Vehicle just means normal. This is control, this is normal. And here's those regulatory factors, right? And you introduce an mTOR inhibitor and you knock those things out, you knock them down. You abolish some of that signaling. And so mTOR ends up being central to all of these forms of anabolism, even if we get out of canonical mTOR signaling. And we're going to do something with like steroid hormones. All right, these are hormones made from cholesterol. Most of your hormones are just a bunch of amino acids. Steroid hormones you make from cholesterol. They don't have cell surface receptors to bind to, like an insulin would, like a growth hormone would, or a glucagon, or an epinephrine, or whatever. Steroid hormones just go straight into the cell. But even with steroid hormones, there's a ton of interaction with cell signaling. There's a ton of mTOR interaction. And so people who want to get hypertrophy in response to training and who aren't getting it have a shortage in mTOR signaling. And that can be remedied. Just like when we were talking about fat loss, there are reliable ways to accelerate fat metabolism. A lot. You can really accelerate fat metabolism. You have to be cautious in how much weight you're losing every week. But there are reliable ways to interact with lipolysis and beta oxidation, reliable ways to accelerate this. Same thing with growth. There are reliable ways to make your tissue grow and get huge way faster. So cell signaling, what is cell signaling? We've been talking about it for most of the semester, right? So epi or norepi binds to a beta-adrenergic receptor. We're going to activate adenylate cyclase, adenylate or adenylol, it's adenylate here. That converts ATP into cyclic AMP. Cyclic AMP, second messenger, right? First ever discovered second messenger molecule, binds to activating PKA. PKA phosphorylates stuff. PKA phosphorylates tons of things. What are the things that PKA phosphorylates? Perilipin. Was that you? And who said the next thing? Was that you? What would you say? Hormone sensitive lipase, a perilipin, hormone sensitive lipase. We're out of markers.
Are they in that box?
Is there? Oh, okay. Oh, there's one right there. Alex, what are your three? Yeah. What is team three called? I can't remember. Okay, you're racked. Alexis, you are? Okay. The sixth. So PKA phosphorylates a ton of catabolic things, way more things than that. But those are a couple of the things that is going to get your calories mobilized, get your energy substrates on the march. Now, insulin is going to obstruct PKA signaling out. PTP. So I think without like, Cameron, did you mumble something too?
Well, they said that I didn't get it out.
Okay. So PDE, phosphodiesterase. So what does phosphodiesterase do? Yeah, 1 through 5. So PDE is going to convert cyclic AMP to regular AMP. Regular AMP doesn't bind to PKA, so PKA isn't activated. So all the things that PKA was going to do, it is now off work. It like clocked out. It was put on leave. So like, oh, okay, so its task doesn't get done. Now, insulin is going to do a lot of other things, not just come in and mess this up with PDE. One of the things that we know insulin does, you're going to go through PI3K, phosphatidylinositol 3 kinase, and then PDK is actually, these arrows are a little bit, so phospho inositide dependent kinase 1. This is the thing that actually activates AKT or PKB. But PI3K phosphorylates PIP2, it becomes PIP3. PIP3 provides this docking station for PKB, so PDK can activate. So there's a cell signaling cascade. A bunch of events take place. And downstream from AKT or PKB, downstream from that, phosphorylating a ton of things, just like PKA phosphorylates a ton of things, PKB phosphorylates a ton of things. One of those functions is to get GLUT4 translocated, glucose transporter 4 translocated. Let's bring some sugar into the cell. So this is just cell signaling, right? This is insulin right here. It's 51 aminos over here. This is a cell surface receptor, right? So we have a phospholipid bilayer. If you're a protein, you can't just go right in. Thyroid aside, thyroid is sort of an exception. And for everything else, if you're an amine or a peptide or a protein hormone, you can't just go right into the cell like a steroid hormone could. You have to bind to a cell surface receptor. In the cell surface, there's a little door. You have to go and knock on the door and say, will you deliver my message indoors? That's what you have to do. If you're insulin, if you're glucagon, if you're any of those things, you have to knock on the door and deliver your message. And you deliver it in a way like this. Step after step after step. A lot of these are phosphorylation processes. So hypertrophy, just like atrophy, is degradation outpacing synthesis. That's what atrophy is. And if you want to lose fat, you just have to burn more fat than you deposit. Right? You have to consume, your body metabolically consume more than your mouth consumes. If reesterification and deposition of fat are outpacing its degradation, are outpacing its oxidation, then you're gaining fat. If oxidation is outpacing accumulation, then you're losing fat. Same thing with muscle. With skeletal muscle, tons of protein in skeletal muscle. And this balance, protein turnover, this balance between protein synthesis and protein degradation. So atrophy is degradation outpacing synthesis. Hypertrophy is synthesis outpacing degradation. And this is the genetic expression difference. This is mostly what your genes are deciding to do. But it can go in either direction. You'll see some people just do whatever they can to gain weight, and they just keep losing muscle and losing muscle. Other people are just go to the gym like a few times, just sort of occasionally show up, and their muscle seems to be growing. This is just synthesis. The relationship between synthesis and degradation determines all this. Now, what proteins are we talking about? Earlier, I said the sarcoplasmic accumulation of proteins. There's a ton of proteins in skeletal muscle. Tons of proteins in skeletal muscle. It's not all just actin and myosin. You got to hold that actin and myosin in place with stuff. There's a bunch of linkage proteins, dystrophin, stuff like that, holding all this stuff in place. Alpha actinin is holding your actin in place, adhering it to the Z disc. You have desmin, you have titin. Titin, the longest known protein in the world. It goes from the Z disc to the M line. In the M line, you have myomesin, you have M protein. There's proteins all over inside of a sarcomere, all over inside of muscle cell. And you're going to be depositing a lot of these things. And it depends on the nature of exercise, which of these things you are increasing the most. Now we're going to get to stuff like integrins, which are integrating the extracellular matrix, this space right outside of the cell, integrating mechanical stresses in this space inside of the protein. You're sensing mechanical loads. But the way that resistance training is going to stimulate synthesis is just cell signaling cascades. Cell signaling cascades from a few different directions. One of them is hormonal. What's your endocrine system doing in response to exercise? Your endocrine system, and autocrine and paracrine, your hormones that are released from stuff. Mostly we think of hormones as released from glands and going out into systemic circulation. That would be the endocrine proper hormone. Stuff like insulin, it goes everywhere. The pancreas is named after going everywhere. Pan, what does pan mean? All, yeah, all inclusive. What is that you? Pardon? And the thing, okay. Did this accept? Yeah, okay. So panoramic, right? Panacea is cure all. A panoramic is again like visualization of everything. Pandemonium, house of all demons, pan demon, right? Pan means everywhere. Creus means flesh, the organ of all flesh, meaning insulin just gets out of systemic circulation. That's all it means. So those are endocrine hormones, is out in systemic circulation. But other stuff makes hormones too. Pretty much everything makes hormones. Like pretty much every cell is going to make some sort of hormones. We don't think of the liver as a gland. We think of it as an organ, right? But that's where your IGF is mostly coming from. IGF is also coming from skeletal muscle cells. Skeletal muscle itself is releasing IGF. We don't think of our stomach as a gland. We think of our stomach as an organ. What comes out of our stomach? Ghrelin. Ghrelin comes out of the stomach. What about fat? What is fat releasing? Leptin. Is that your thing? So all, wherever you are, it just doesn't matter where you are in the body. All of these cells are releasing hormones. Some of them are endocrine. Some of them are paracrine, meaning immediate neighbors. Some of them are autocrine, where you just, some of them are intracrine. But the autocrine means like if I'm a cell, the best way that I could describe it is, because you have to release it and then it binds to a cell surface receptor. So it's inside of you, it comes out, and then it binds to a cell surface receptor. So it's as though a cell farts and then smells it. It used to be inside and it wasn't causing any sensation. And then it became outside and then it starts like binding. That would be autocrine signaling, where it actually does get released. But in all of these cases, intracrine aside, in autocrine and paracrine and endocrine signaling, we have a little compound that binds to a cell surface receptor. And we just think of it as some little, let's just say it's a hormone, a ligand, a hormone, or whatever, binds, it turns something on, right? And then that turns this thing on. And then that thing turns that on. And then that thing turns that thing on. And then that thing does something else. These cell signaling cascades. And so this is the equivalent of like a Peanuts character in terms of its level of detail and its artistry. But cell signaling cascades, it's just insulin signaling, right? It's beta-adrenergic signaling. All of these different signaling cascades, most of them function as a relays. Now, if the gun goes off, which in this case, the starter pistol means something has just finished binding to a cell surface receptor. Let's say it's epinephrine, has just finished binding to a beta-adrenergic receptor. At that point, that's the starter pistol, okay? The first runner goes, you don't send all the runners at once. You're not like, okay, the starter pistol goes off, every single runner start running. Because then only the anchor matters at that point. Why even have the other one? So the first run, the starter pistol goes off, the first runner does her thing, right? And then passes the baton. This is like a phosphate. We're going to phosphorylate this runner. As soon as the phosphate is attached, this runner's active. This runner's going to go activate another runner. That runner's going to activate another runner. That runner's going to activate another runner. It depends how many runners there are in the relay. But eventually, one of these runners crosses the finish line. And it depends where the finish line is. There could be a number of responses. It could be GLUT4 translocation, could be the finish line. It could be genetic expression, could be the finish line. right? There's a number of finish lines. It could be like tumor suppression or something. Maybe this is one of the, like a P53 or something like that, where we're regulating cancer. There's a number of finish lines that this could be, but cell signaling is like a baton race. So name a few things that it could be. Insulin, glucagon, epinephrine, IGF, growth hormone, integrins, interesting, yeah. Yeah, so mechanotransduction, integrins would, maybe it wouldn't be like binding in the same way, but you're gonna initiate that same thing with integrins, yeah. Lots of stuff is going to bind and initiate the signaling cascades. Now we know this one. We know the interaction between PKA and PKB. All right, here's your beta-adrenergic receptor. There's adenylyl cyclase, which converts ATP into cyclic AMP. Cyclic AMP binds to PKA. PKA is a kinase. It phosphorylates stuff. There's hormone-sensitive lipase. There's pyrrolipin. Now we have glycolysis over here. There's insulin binding to an insulin receptor substrate. There's PI3K. You see PIP3. PI3K is a kinase that is going to phosphorylate PIP2 and make it PIP3. Phosphatidylinositol triphosphate from bisphosphate or diphosphate, whatever you want to call it. PIP3, this is our docking station so that PDK can activate PKB. PKB then phosphorylates lots of things. Before we were focusing on PDE. This is a fast cell, so it's PDE3B. Remember, there's a bunch of kinds of PDE. What is the thing that most of you had today that's a very weak PDE inhibitor? Caffeine. Who said it first? I don't know. Who said it like in tandem? It's hard to tell with masks. Because usually I hear sound, I look in that general direction, I see lips moving. Whatever, caffeine. So methylxanthines are weak PDE inhibitors, but what we've been focusing on all along, because we've been talking about lipolysis and weight reduction, is PDE, this relationship where if you have active PDE, you don't have lipolysis. So you want to prevent PDE, and how do you prevent PDE from being activated? Well, methylxanthines will help a little bit, but man, let's not have insulin because that's the most powerful inhibitor of lipolysis that we know of, is insulin. But PKB has other functions too. PKB gets into mTOR. There are other functions as well. And if we look at, remember, AKT and PKB, exact same thing. They're just different words for the exact same thing. So PKB is deactivated. It's inhibiting tubrin. TSC2, tuberous sclerosis complex 2. That's tubrin, tuberous sclerosis. It's a condition, you can look it up, these sort of out of control tumors. If you don't have regulation of the tuberous sclerosis complex, you get tuberous sclerosis. But TSC2, that is what PKB is inhibiting, is TSC2, tubrin. Tubrin. Now, TSC2, it looks like it's doing this. That's not quite how it works. It's doing this. So tubrin is inhibiting RHEB, R-H-E-B, ROS homolog enriched in brain, RHEB. RHEB is a direct activator of mTOR. So PKB is inhibiting tubrin. Tubrin would be inhibiting RHEB. RHEB would be activating mTOR. So if you shut off the thing that's shutting off the thing that's activating that thing, what does PKB do to mTOR? It's activating it, right? Two negatives. Two wrongs make a right. Two negatives make a positive. So you shut off the thing that's shutting off the thing. If like I'm about to go shut off the light, and I don't know, this is stupid. It analogy. I'm not going to finish it. But you get the idea. Where if there's something that's policing, right? Tuberin is policing this promoter. AKT just shuts off the police. And suddenly the promoter is able to carry out its action. So once you activate PKB or AKT, that allows mTOR to be activated. Now, mTOR is just an enzyme. mTOR, mammalian target of rapamycin, mechanistic target of rapamycin, whatever you want the M to stand for. You can make up something new. My target of rapamycin. This is halting. It's a kinase. It phosphorylates stuff. mTOR, the enzyme, is a kinase. Kinase is phosphorylate. I think there are phosphatases. What does a phosphatase do? You're working through it correctly.
Enzyme phosphorylates phosphate?
It removes the phosphate. Yeah. It's going to remove the phosphate. What do you ract? Okay, ract. 30 seconds. So like PIP2 to PIP3. What is the enzyme that converts PIP2 to PIP3? Yeah, PI3K. So PI3K. P10 is going to convert it back. It's a phosphatase. So it will convert it back. So there are phosphatases and there are kinases. A lot of these cell signaling cascades are working with kinases where you phosphorylate things. But when you phosphorylate something, you can activate it or deactivate it. It depends on the nature of that phosphorylation. So mTOR is a kinase. It phosphorylates stuff. 4E-BP1, this is inhibiting protein synthesis. So you inhibit the inhibitor. You phosphorylate 4E-BP1 and shut it off. It is an inhibitor and you have now shut it off. P70S6K, sometimes they'll just be S6K or S6K1. You'll see this in a number of ways. Most commonly probably is P70S6K. This is the promoter of hypertrophy, of protein translation, P70S6K. mTOR turns that one on. So it shuts off the inhibitor and it turns on the promoter. Just like glycogen synthase. If you are insulin, you're going to bring carbs into the cell and turn on glycogen synthase. If you are PKA and you're trying to mobilize carbohydrates, if you're PKA and you're trying to do glycogenolysis, glycogen lysis, we're trying to break up our glycogen and get a bunch of carbs on the march, you're going to shut off glycogen synthase and turn on glycogen phosphorylase. It's not enough that you turn on glycogen phosphorylase. You have to shut off glycogen synthase at the same time. So you turn off your opposition and turn on your promotion. So that is what mTOR is doing. It's shutting off opposition for EDP1 and it's turning on promotion so that protein synthesis can follow. Now P70S6K, it's another kinase. That's what the K stands for, kinase. RPS6, ribosomal protein S6. That's actually what gets phosphorylated. Ribosomal protein. Where does translation happen? Ribosome. Protein translation is not happening in a nucleus. That's transcription. Transcription is in play. Translation, where you're actually making proteins, linking together amino acids one after another until you've manufactured a protein, that happens in a ribosome, which is not in a nucleus. And so when you see this phosphorylation of ribosomal protein, S6, now we are really close to protein synthesis. So that's the last few steps in the directions, right? If you're going to drive from here to, what's a destination? Where do you want to go? Canada. If you're going to go from here to Canada, which is probably on fire too, then like your MapQuest or your iPhone directions or whatever, the last few steps are going to be these right here before protein synthesis. But there's a lot of steps before, and there are other steps after as well. There are side roads. PKB is busy. PKB does a ton, bless you, of stuff. I like the double cut. The armpit and the mask were all safe. And we're going to talk about some of these things. But it gets busier. It gets even busier than that. I mean, it looks, at this point, it's starting to look hairy. It looks like it has fur or something coming off. That's AKT, probably can't read from where I can barely read it from here. But all the stuff coming in, all the stuff coming out, that's not even all of it. PKB has an enormous amount of functions, but those functions are anabolic. PKA is similarly busy. PKA is similarly busy with catabolism. PKA is catabolic. PKB is super anabolic. And it's nice to call them PKA and PKB because they really are counterparts to one another. Saying PKA and AKT doesn't illustrate it on grounds of nomenclature, their sort of brethren status. So AKT or PKB though, again, same thing. This is just named after, we always name stuff after its discovery, either after its discoverer or after the disease, tuberous sclerosis complex, or in this case, after a mouse with thymoma, this is the T stands for thymus cancer. And so we just name stuff at the time, not realizing how important these things will be later. mTOR went under a bunch of different names. Raft, FRAP, and then mTOR was the one that stuck. But in some of the early literature on mTOR, it's RAFT or it's FRAP, F-R-A-P. And eventually we settled on mTOR. But in all of these, the RAFT with the F, FRAP begins with an F, FKBP12, this is where rapamycin works. Mammalian target of rapamycin. So we're all talking about rapamycin in these names. Rapamycin is not an endogenous chemical. It was discovered on Easter Island, the island with all the faces, those big stone faces. Rapa Nui, is the language. Rapamycin is the chemical. And so we named it this thing like mammalian target of rapamycin, not realizing that it was sort of the hub of all metabolism. I think we would have come up with a more precise name for what it is, metabolic enzyme omega or whatever. We would have come up with something slightly more honorable, I think, than mTOR. But all of these names, it's just at the first discovery, we don't know how important some of these things are. Some of them turn out to be not important at all. Some of them turn out to just be world changing. This is one of the medicine changing, exercise changing, everything changing enzymes that was discovered. So here's PKB. PKB kinase, right? Protein kinase B. And so you see these little P's right here. Well, these P's are not just going to AS160 AKT substrate of 160 kilodaltons. It's molecular weight. So often when you see numbers in these, it's just molecular weight. And so AKT substrate 160. It's phosphorylating everything. I mean, it's a kinase. That's what it does. It phosphorylates stuff. But the image makes it look like it's only phosphorylating a couple things and it's doing something else to the other stuff. That's not how it works. But so these are the main functions of PKB. Glucose uptake. Okay, AS160, again, AKT substrate with 160 kilodaltons. This thing is inhibiting GLUT4 translocation. So you inhibit the inhibitor. All right, now what happens to GLUT4? Yeah, it moves, right? Quick form moves. Said nobody, but I think everybody thought. That's the glucose uptake. If we go over to the metabolism part, you see GSK3 and another inhibition there. Glycogen synthase kinase 3. That's not glycogen synthase. That's glycogen synthase kinase. This is actually shutting off glycogen synthase. So again, turn off the thing that's turning it off. Inhibit the inhibitor. We know FOXO, forkhead FOXO. We've talked about that from the perspective of cancer. Remember the AMPK is turning that thing on. Bad. This is like getting into cell survival. Degradation, cell survival. We can put FOXO is in there as well. This blue line survival, all of this stuff. BLC2 associated agonist of death. That's what BAD stands for. BLC2 associated agonist of death. Blah, 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 agonist of death. Bad. And then the caspases, CASP ACE. This is caspase 9 over here. So this is degradation. This is atrophy. This is apoptosis. Over here, this is where wasting away. And PKB is shutting off all of these things. So CASP is cysteine aspartic protease. CASP, cysteine aspartic protease. That's number 9. That means there's 1, 2, 3, 4, 5, 6, 7, and 8. There also happen to be 10, 11, 12. There's a bunch. There's a bunch of things. The proteases, what do the proteases do? Break down proteins. Yeah, the protease garbages up proteins. Endothelial nitric oxide synthase, enos. Remember that if you have sort of an anoxic experience or a hypoxic experience, we're getting signals released. We're sending signals for adaptation. The response is we don't have enough oxygen, right? If this is hypoxic, we don't have enough oxygen. Hypo, low oxygen. And so you'll release vascular endothelial growth factor, VEGF. You'll activate this. You'll activate that. And you'll make some new vascular roadway. So this is through mTOR signaling. Unotubrin, TSC2. Tubrin. What does tubrin do?
You inhibit it when you turn on oh man, PKB, and then you inhibit tubrin and you inhibit red, which turns on mTOR.
Yeah. So it's a negative regulator of hypertrophy. Pras40. Again, the names of some of these things don't matter. Proline-rich AKT substrate, 40 kilodaltons. This is another negative regulator of mTOR. That PKB is inhibiting the negative regulators of mTOR. Proliferation. This stuff, when we talked about ubiquitin proteasome system, remember that there's hundreds of E3 enzymes. The E3 that actually does ubiquitination. You attach the protein and there's like 600, 700, 800, I don't know, there's hundreds of these E3s. There's one of them, MDM2. It's like, you can look up what that stands for. It's something like a mouse minute to homolog, whatever, mouse something, whatever, who cares? This thing, it's a type of E3. And what it's ubiquitinating, what it is breaking down is P53, not P27, but P53. P53 is a protein. What do you think its molecular weight is? 53 kilodaltons. So it's degrading P53, and P53 is a tumor suppressor. So PKB turns out to be this horrible thing if you have cancer. Again, this is so P53, P27. These things are P53 has this name. It's like defender of the genome or something like that. People call it these things. It's such wonderful proteins for the regulation of cancer. But PKB just says grow. Suppressors of growth need to be suppressed. So that's what PKB does. It is super busy stopping programmed cell death and protein degradation, stopping, sort of managing tumors, stuff like that. It is trying to promote the accumulation of carbohydrate over here. It is, let's create new vascular channels. Let's turn on mTOR for protein accretion, protein growth. Let's get sugar into the cell. It has so many functions, and all of them are anabolic. All of these functions for PKB are anabolic, or at least anabolic adjacent, right? They're doing something that is supportive to the sort of generalities of anabolism. So that's PKB. Remember, PKA, everything PKA does is just break down catabolic. Everything PKB does is build up. So if you are working with a patient and you're like, the oncologist has sent this patient to you. Let's say you're doing like cardiac rehab, and an oncologist sends you a patient. Do you want PKA on or PKB on? PKA. You want to block PKB the best you can. Now, let's say somebody comes to you who's like 20 years old or 25 years old or something, and they want to do bodybuilding, and they're in like their mass phase. PKA or PKB? PKB. You want as much PKB signaling as possible. Okay, so let's go right there. PI3K is going to turn on. It says AKT, but right, PKB. Again, tons of stuff all over the place. Cytokines, little proteins, little signaling proteins that are released from cells, cytokines. Those things are either pro or anti, right? These signaling proteins are going to signal, a lot of these are going to signal mTOR. IGF-1, right? We're going to have our signaling cascade with IGF-1. More inflammatory stuff like interleukins, and there's a bunch of different interleukins. Growth factors, extracellular matrix, so there's your integrins. And over here we see WNT, W-N-T, WNT proteins. What? At the beginning of the lecture, what was the context in which I mentioned WNT proteins? Satellite cells. Whose mouth was? Okay, I should recognize your voice. So satellite cells, WNT proteins. This is part of the. I have no idea what it is. The one. WNT proteins are part of this satellite cell activation. If we're going to have hypertrophy owing to satellite cells, WNT protein signaling. Now, this is a really good article. It'll be uploaded later today, but these WNT proteins increase mTOR complex 1 activity through the inactivation of tuberin. So WNT proteins, they have their own frizzled, which goes to disheveled, whatever, but frizzled is the cell surface receptor that they bind to. And we're activating mTOR through those WNT proteins. So a lot of this gets relayed through mTOR. It's not as though mTOR is the only road to hypertrophy, but nearly all roads, not all, but so many roads converge at mTOR. It is such a critical hub. I mean, there's so much going on here, but this PI3K to PKB to mTOR, that is I-5 going from Mexico to Canada. That's I-5. This is the main way if you want to go up there. Sure, there's scenic routes and stuff. But so here's PKB. There's tuberin. This one has Reb. So tuberin would be suppressing Reb. Reb would be directly activating mTOR. There's Crash-40, we talked about that one earlier. It was in one of those, is that negative regulator. Proline-rich AKT substrate of 40 kilodaltons, whatever. Raptor. Raptor recruits downstream targets. So the mTOR, the kinase, can phosphorylate. If you don't have a Raptor, mTOR is dysfunctional. mTOR cannot permit growth, cannot permit protein translation, will not turn on the end of that cascade without Raptor. So Raptor is critical. Deptors, we'll talk about this one. That's much more fascinating than Crash-40. Deptor is another negative regulator, but it also has anabolic signaling as well. There's FOXO over there. So FOXO, PKB is shutting that off. Over there, you see GSK3, glycogen synthase kinase. It's shutting that off. Glycogen synthase kinase would be a tuberin promoter. So PKB is shutting off glycogen synthase kinase so that we can do glycogen synthase, and it would be promoting that, and it's directly inhibiting that. Remember, downstream from mTOR, it looks like these things are coming from Deptor. It's not. That's just the diagram. Downstream from mTOR, the kinase, we're phosphorylating. That's what these little P's are right here. These little P's. S6K or P70 S6K or S6K1, whatever you want to call it. That's a kinase which phosphorylates S6. That's a ribosomal protein S6. And it is inhibiting 4-EBP1. 4-EBP1 is that negative regulator of translation. And remember, translation is at the ribosome, the linking together amino acids, the synthesis of a protein, and the synthesis of proteins is hypertrophy. If a muscle is to grow, it is through the synthesis of proteins that it is growing. It is mostly through mTOR. Negative regulator translation, it's inhibitor. It's actually not direct, but it's pretty close to direct. Directly inhibiting regulation, translation. So again, cell signaling cascades. These cell signaling cascades, they end somewhere. And we began the lecture looking at the relay race, the baton relay race, and thinking phosphorylation cascade. Attach a phosphate to something and it activates it. In that case, activates it. Where it ends in the nucleus is commonly where cell signaling cascades end. But again, not always. You don't have to end in the nucleus. That's just where things commonly end. It could end at the ribosome. It could end with GLUT4. There's a lot of endings for these things. But transcription is in the nucleus. Let's make our RNA. Let's make the recipe that we're going to take to the kitchen to cook up a protein. So your RNA is just, it's a recipe. And you take it to the kitchen, to the ribosome, and say, hey, make this dish. And you link together all the right amino acids to make that dish, whatever it is, eggs Benedict or whatever. That's translation, is using a recipe in the ribosome, in the kitchen, in the cell's kitchen, not in the nucleus. Using that recipe to cook up a protein. So that would be translation, and increasing translation. That this is mTOR's big role. Let's increase the amount of dishes we're cooking up. Now there are two mTOR complexes. There's complex 1 and complex 2. Complex 1 is the one we talk about. Complex 1 is hypertrophy. Complex 2 has a bunch of functions. Complex 2, ion regulation, stuff like that. Complex 2 also activates complex 1. But they have different downstream targets. So here's complex 1. Regulatory associated protein of TOR, that's Raptor. Richter, rapamycin insensitive companion. Raptor this is really what defines complex 1, right? We have Raptor. See, MLST8, sometimes it's called Gable, G beta L, Gable. So you'll see it written both ways. G protein beta subunit like, or MLST8. It doesn't really matter. This is the least important protein here. That's in both of them, Gable or MLST8. mTOR, that's in both of them, that kinase. Raptor is in complex 1. Richter is in complex 2. Important for the identification of how we figured out these things. What does mTOR stand for? Mammalian target of rapamycin. So when we see Richter, rapamycin insensitive, what does that mean? It doesn't affect it acutely. It doesn't affect it. Chronically, it does. The assembly of the complex. So if you take it chronically and just keep taking it, keep taking it, keep taking it, eventually you'll suppress this. But acutely, it doesn't affect it. Acutely, rapamycin totally knocks this one out. So that's really how you tell the difference between these two things. Give someone rapamycin and this is gone, and it takes chronic administration of rapamycin to do anything without it. But downstream, you see 4-ABP1, S6K1. It's been different on every single slide. P70, SSK, SSK1, whatever. So that's downstream here. AKT, you see downstream from complex 2. So that's PKB, right? So that's upstream from complex 1. So if you activate complex 2, downstream from that, you actually activate complex 1. Protein kinase C, SGK. So there's other stuff that's involved, ion regulation, stuff like this, is what those things are doing. Complex 2 has a ton of functions, but complex 1 is what we focus on. And some of this stuff is just recruitment. We're going to talk about Depter later. These ones are just for recruitment of downstream targets. Just like Raptor in complex 1 helps recruit the downstream targets so that the kinase can phosphorylate them. So the mTOR kinase is capable of phosphorylating those downstream targets. You have these necessary for recruitment of downstream targets. Now here's the connection between the two. Complex 1, right, you see mTOR, MLST8 or Gable, and Richter. Downstream, you see PKB. PKB shutting off tubrin. Tubrin would be shutting off Rev, and so mTOR can be activated downstream of complex 2. If you rely on complex 2 to do all of your complex 1 signaling, you will have a fatal shortage of complex 1 signaling. You need other stuff to activate complex 1. Namely the IGF doing a ton of this signaling. But lots of things are going to interferon gamma and interleukins, a bunch of 2 and 15, especially these interleukins. And, there's just a ton of Wnt proteins, and then obviously IGF and insulin. And there are all of these promoters of mTOR complex 1. mTOR complex 2 is one promoter, but you will be frail and then gone if this is your entire source of mTOR complex 1 activation. So a bigger look at all of this stuff, and a lot of this will make sense too. And some of it you'll already recognize. BAD, right? We talked about that one, that BLC2 associated agonist of death, glycogen synthase kinase 3. There's glycogen synthase, there's glycogen synthesis. PKB, there's tubrin, you see ANPK. LKB1, liver kinase B1, that's a promoter of ANPK. We'll talk about that later. You see Rev and mTOR, there's Cras40, P70, SSK, 4-ABP1. A lot of this stuff, there's mTOR complex 2, there's PDK, right? So we're going to activate PKB through PDK. There's PIP3, so there's an insulin receptor. There's PI3K, convert PIP2 to PIP3. There's P10, which I said takes it apart. So a lot of this stuff is at least recognizable so far. And we'll start going through more and more of it. It is a nice diagram with all of these, explaining what the colors are, explaining what it means, if it's direct stimulation, direct inhibition, there's something indirect going on. And then just a closer look at PKB and everything that it's doing, how busy that thing is. But the narrative summary so far of mTOR is that PI3K gets activated. Phosphatidylinositol 3 kinase, it gets activated. Lots of stuff can activate it, but something does. You phosphorylate PIP2. Phosphatidylinositol bisphosphate and now it's triphosphate. PIP3 docks the PKB so that PDK can activate it. Phosphoinositide dependent kinase. That can now activate PKB. PKB does tons of things. PKB just goes everywhere. But one thing it does is inhibit tubrin, TSC2, tubrin. Normally, tubrin is shutting off red. Normally, tubrin is shutting off red. Once you shut off tubrin, red is allowed to activate mTOR. mTOR phosphorylates its downstream targets because mTOR is a kinase. It phosphorylates stuff. It phosphorylates its downstream targets. P70SSK, 4EBP1. 4EBP1 would have been inhibited protein translation, but now you shut that off. P70S6K phosphorylates ribosomal protein S6. You're doing translation at the ribosome. Let's start translating. So that's the narrative summary of mTOR. The briefest way of talking about it is stuff affects mTOR. mTOR is a major signaling hub inside of your cells. It's not just muscle, it's all over the place. If you want, when we get to the brain and exercise, we'll be talking about mTOR. mTOR phosphorylates stuff. It's really close to the, it's not, if we're gonna drive, let's do the drive from, figuratively, do the drive from Canada to LA. Okay, so the first major city is what, Seattle? Coming from Canada, to Seattle, and let's ignore like Tacoma because it's Seattle. And then Portland, I guess, would be the next one. I would say Salem because that's my hometown, but if we're being honest, we can just clear the rest of Oregon. Maybe Eugene, maybe. Yeah, every other town, you go through like Meade and whatever. And then what's the next major town? Sac? Reading, yeah, Reading or Sac. And then I guess Stockton, if you have to. Just put on your Kevlar if you're gonna get gas in Stockton. And then you keep going south, what's next? LA. And then San Diego, and then you're there. There are some major stops along the way. And so you can say PKB is Seattle or something. mTOR is like, that's LA. mTOR is LA. You're not quite to the border, but that is a huge metropolitan, that's like capital of the West. This huge metropolitan like conglomeration of traffic. You're pretty close to the destination. You're not quite there. There's a little bit more to do. You still got San Diego and Oceanside left to go. But mTOR, you're getting really close. This article will be uploaded. David Sabatini is probably the single best researcher on the subject of mTOR. One of these founders, there's a few people you could point to and say founder. He's one of those few. Some really good work. But it says mTOR to glance, and then it looks really complicated. So it's maybe a little bit more than like a glance. But it's a very effective article. So we'll stop here at our checkpoint. What is mTOR? Mammalian target of rapamycin. And what is an mTOR complex? Yeah, there's a bunch of proteins all linked together. How many mTOR complexes are there? There are 2 mTOR complexes. How does mTOR signaling work? These cascades, it's a bunch of phosphorylation. Mostly, it's mostly a bunch of phosphorylation. What does mTOR signaling accomplish? Yeah, translation for mTOR complex 1 is a bunch of protein translation. Okay, that's it for the day. On the board. I'll make a list of some possible test questions. The test is coming up. I think maybe we'll delay it a couple of days so we have enough time to get through mTOR. So it's not going to be when it says.
So not on Monday.
Oh, this coming Monday?
Yeah.
Okay, let's delay it a couple of days. Let's say like Friday or something. So we have enough time to get through mTOR.
Are we gonna have like maybe like a review session?
Sure. Yeah, we'll plan later though.
Okay, yeah.
But I'll come up with a list of a few questions, maybe like 5 or 6 questions or something. And I'll give them to the teams just so you can see what the test is, level of detail that's going to be on the test, so you can be ready for it. So it's not just a huge surprise when the test hits you like the Monday after a break. So I don't know, I'll get that done this week at some point, and I'll pass them out. And then it'll just be a little extra credit thing, where each team fills it out and submit those, and each question correct will be a little bit extra credit. I haven't been thinking. Yeah, I plan on thinking about that. What I was imagining was a couple of case studies. Obviously, it's going to be like a weight loss case study. It's going to be a weight gain case study, and it's going to be like maybe a kid case study, something like that. A few case studies where you can do the risk factors and risk stratification and an exercise prescription for that particular population. Then maybe some case study that has cardiovascular disease or diabetes or something like that. So maybe like 4 case studies, and then just some multiple choice questions. Maybe what we'll do is but to do that, we all have to be live. We have to be in person. And there's a couple of people who are waving at them now. Their only engagement with classes. I'm waving at the camera. So that's why it's a little bit hard to figure out. Because what I would like to do is just like even the multiple choice, just sort of circle it as opposed to click somewhere online and stuff. So I'm not exactly sure how I'm going to structure the test. What I like to do when I do mTOR, some of you are listening to mTOR, like, yeah, I said this before, because you've had me in a previous class where I talked about mTOR. Other people are sort of like brand new to mTOR. There's tons of vocabulary, and it's like you've just stepped into German 4 without doing German 1, 2, and 3. And so I will still go at a understandable pace, even though there's half of the people have had me before. But usually what I do is I just have a huge piece of paper and I say draw it, get everything possible of mTOR onto this sheet of paper, and let's see how much you remember. Hard to do that with the format today. So that was an unhelpful answer, but it lets you into my unthought out thought process. Okay.
How do you researchers study mTOR activity?
A lot of inhibition, which mTOR complex 2 is a little bit harder to inhibit, so it makes complex 1 easier to study. But in most of these things, if you inhibit it and notice the downstream, like, oh, this stopped happening, then even like studying mTOR in satellite cells, we just abolish this and see what happens. And so you want to investigate some particular physiological phenomenon. You need to start inhibiting things and seeing what happens to that phenomenon. So inhibition tends to be the best way to study the effect or the contribution of some enzyme